This video will explain some of the important research that led to our current understanding of DNA, its structure, and how it replicates. So we're going back to the 1950s. Back then, we knew that DNA was a thing, but we weren't really sure what it was for or what it was like. We just knew that there was this goop you could extract from cells that was DNA. And one of the big debates was whether DNA was the genetic material, the material that was passed on from parents to children, that contained our genetic instructions, or if it was protein. A lot of people thought protein seemed like a more likely candidate because proteins were very complex and varied. They came in a lot of different shapes and sizes, and so they thought something like that would probably be what would hold the genetic material. DNA seemed like it was kind of simple, like it was sort of all the same shape, and they didn't think there was that much variety in it. This first experiment didn't directly show that DNA was the genetic material, but it sort of paved the way for the next experiment, which did show that DNA was the genetic material, and this is the Griffith experiment. So there was this kind of bacteria, and it came in two varieties. There was the rough strain and the smooth strain. And we can see here that the rough strain was harmless. The mice that were injected with the rough strain of the bacteria survived. The smooth strain had this kind of extra coating on it, and when the mice were injected with the smooth strain, they died. Now, the experiment, what they did was they also tried killing the smooth strain with heat, and when they did that, they found that the mouse survived. Then, this was the key part right here, they put in a mix of the rough strain, which is usually harmless, and the smooth strain that was dead. And they expected that the mouse would survive because the rough strain didn't kill the mouse, and the smooth strain that was dead also didn't kill the mouse. But what they actually found was that the mouse died when it was injected with this mix. And so what they realized was that the smooth bacteria that were dead, something in them was getting transferred to the rough bacteria, and it was turning the rough bacteria into smooth bacteria. And once that happened, they killed the mouse. So there was something transferring from the dead bacteria to the live bacteria. And they knew whatever was transferring, that had to be the genes, that had to be the genetic material, because that was allowing that bacteria to change. And they called this process transformation. Now we come to the Avery experiment, which kind of piggybacked off of the Griffith experiment. In the Avery experiment, they said, okay, whatever is transferring from the one bacteria to the other bacteria, that is the genetic material. So what if, one at a time, we destroy the different kinds of molecules in the bacteria, and then we see which one is actually causing the transformation. So one by one, they destroyed the various molecules using enzymes in the bacteria. So they tried destroying the polysaccharides, and they found out that the mouse still died. So the thing that was the genetic material wasn't the polysaccharides. They tried destroying the lipids. The mouse still died. So the thing that was transferring the genetic material wasn't the lipids. They tried it with all the different molecules, including protein, and when they destroyed the protein, the mouse still died. So if protein was the genetic material, the mouse should have lived because those dangerous genes wouldn't have transferred. But what they found is when they destroyed the DNA, the mouse lived. The bacteria did not transform. And so that showed them that the genetic material, the thing that was transforming the bacteria and giving them new genes, was in fact the DNA and not anything else. The Hershey-Chase experiment is another experiment that provided further evidence that DNA and not protein was the genetic material. In the Hershey-Chase experiment, they used bacteriophages, which are small viruses which infect bacteria, and they realized that the way viruses work, they inject their genetic material into a bacteria, and then that's used to make new viruses. So whatever it is they're injecting, whatever molecule that is, that had to be their genetic material. So they designed their experiment to try to figure out, okay, what is it that these bacteriophage viruses are injecting into the bacteria? In phase 1 of this experiment, here shown in red, what they did was they put little radioactive tags on the protein of the bacteriophage. The point of these tags was they could scan later to see where the radiation was, and so they could tell whether the protein went into the bacteria or it didn't. So it's like they attached little radioactive sulfurs all over the protein because they knew that sulfur was found in protein, but it was not found in DNA, so the sulfur wouldn't attach to the DNA. They let the bacteriophages infect the bacteria, and then they separated the infected bacteria and what was left of the virus, and what they found was that their radioactive protein was still left outside of the bacteria. It was not inside the bacteria. It didn't get injected. So that provided some evidence that protein was not the genetic material. That was not the thing being injected. Then they used phosphorus to put a radioactive tag on the DNA part of the bacteriophage, and that's shown in green here. And they ran the same experiment. They let the bacteriophage infect the bacteria, and then they checked to see where their radioactive phosphorus tags were. And what they found was the radioactive phosphorus, which only attaches to DNA because phosphorus is in DNA and not in protein, was inside the bacteria. So that means the thing that got injected into the bacteria was the DNA. Therefore, the DNA was the genetic material. Now that it had been shown that DNA was important, that it was the genetic material, people became a lot more interested in figuring out the structure. How could such a seemingly simple molecule actually contain all of the genetic instructions for making a living being? So Chargaff did some of the research that helped us learn the structure of DNA. He found out that in DNA, there is always the same amount of C as G, of cytosine as guanine. And he found out that there's also always the same amount of A as T, of adenine as thymine. This suggested that perhaps cytosine links up with guanine and adenine links up with thymine, which we now know to be true. So he kind of discovered the base pairing rules. He also showed that the DNA varied from species to species. So there actually was some variety in DNA. It wasn't all the same. Different species had different ratios of A to T and C to G. So one species might have, 51% A and T, and the other species might have, 58% A and T. So that provided some support again that DNA actually might be able to function as a genetic material, since it did vary from one species to the next. Now we come to Rosalind Franklin. Rosalind Franklin was a young woman working in a lab, and she took pictures of DNA using this technique called X-ray crystallography. And right here is her famous picture that she took that really helped show the structure of the DNA. So she was very important to discovering the structure of DNA. Unfortunately, before they could hand out the Nobel Prizes for discovering the structure of DNA, she had died of cancer that she most likely contracted from taking these X-ray pictures in her lab and being exposed to large amounts of radiation. And they do not award the Nobel Prizes posthumously, so it's kind of a sad story there. Now we come to Watson and Crick. Watson and Crick are widely credited with discovering the double helix structure of DNA. They built models in their lab using metal and other various pieces to try to match up what we knew from the research with the actual structure of DNA that would physically fit together. Really, they had a lot of help from some other people, including Rosalind Franklin. There's kind of a controversial story in which they looked at her pictures of DNA and they looked at her lab notebooks. Someone else in her lab let them in to look at that stuff, and they never credited her or any of her work in their discovery. So by all accounts, Watson and Crick were kind of jerks, but they still got the credit and the Nobel Prize for DNA. The last experiment we're going to look at was by Meselson and Stahl. And Meselson and Stahl were really looking at DNA replication, because we know that every time a cell divides, DNA has to copy itself first, so that each cell has a new copy of the DNA, and that DNA continues to be passed on from cell to cell. And they figured there were 3 possible ways this could happen. One way, the conservative model here, said that the original DNA strands, so that's shown in dark blue, stayed together, and then a brand new DNA strand, shown in light blue, was created. So we have one totally original DNA, one totally brand new DNA. The semi-conservative model said that the original DNA split its two strands, and the two new molecules of DNA each contained one original dark blue strand and one new light blue strand. So this was called semi-conservative. Then we had the dispersive model that said that the original DNA sort of got broken up into lots of little bits on either side and mixed with new DNA. So we had these sort of mixed half and half strands. They devised a very clever experiment to determine which of these three models of DNA replication was correct. First, they allowed the DNA to replicate using a so-called heavy isotope of nitrogen. So it was nitrogen that had some extra neutrons, so it was a little bit heavier than normal nitrogen. So now all of our original starting DNA that we're going to start with is heavier than normal DNA, and so we can kind of track where it goes. Then they switched to the normal lighter nitrogen, and so any newly created DNA was going to be of this lighter type. They predicted that after the DNA had replicated one time, if the conservative model was correct, we would have two distinct bands of DNA. We would have a band that contained all the original heavy DNA, and we would have a band that contained all the new lighter DNA. What they found was they did not get two distinct bands like this. They got just one band. So that showed them that the conservative model was wrong. This wasn't enough to show if the dispersive model or the semi-conservative model was correct, because either way, dispersive or semi-conservative, you would expect one band. So they let the DNA replicate one more time. After the second replication, if the semi-conservative model was correct, they expected to now see two bands. And if the dispersive model was correct, they expected to still just see one band because the DNA was all old and new, just evenly mixed. And what they did see was two bands. So this showed them that the semi-conservative model was correct. To summarize, as we look at our three possibilities again, remember that dark blue is the original heavier strands of DNA and light blue is the newly created DNA during the replications. In our conservative model, here are our two bands. We have one heavy band and one light band. However, that did not happen. We only got one band on the first replication, so that one was out. On our second replication, in our semi-conservative model, we get one lighter band and one, these two guys, heavier band. In our dispersive model, we still get one band of the same weight. And what we found was, in our second replication, we got two bands. So semi-conservative was correct, dispersive was out.